Well, maybe we should go around and introduce ourselves real quick since there's so few of us. And then maybe we could get started. I'm Katie Pitts and I work at the Monterey Bay Aquarium Research Institute. And together with I guess five of us here, we're all part of the Better Biomolecular Ocean Practices group. And we're going to give a short presentation going over how that group got started. But a lot of my work focuses on environmental DNA, looking at biodiversity in the Monterey Bay through time. So using environmental DNA as a tool to say how have these communities changed through time and space.
Great, thanks. Yeah, I'm Allison Watts. I'm at the University of New Hampshire. Katie and I have been working on connected on through this project for a couple of years now. Most of my work is working with resource managers in coastal and some freshwater systems, figuring out how we can use eDNA to really support better management. So my work tends to be very, very management focused and a little bit less basic research focused.
Jody Vanderkamp from CSIRO in Hobart, Tasmania, Australia. I'm part of the BeBOP project as well. Most of what I do is DNA-based observations from the environment, a lot of marine work, but also some sort of estuarine, freshwater and terrestrial work as well. I'm part of the Australian Microbiome Project, and I lead the IMOS Marine Microbiome Facility, which has a decade's worth of time series measurements based on DNA. And, you know, for time series data, having really clear methods, SOPs, and, you know, a way to share those SOPs is really important to us. So that's why I was really keen to get involved in this project.
Thanks. Jose?
Sorry, hello, my name is José Luis Moutinho. I work at the Center. I'm the Chief Business and Networking Officer of the Center. I'm helping OBPS with these, workshops and focus sessions.
Thank you. George?
George Cummings, biochemist, a diver, started working with Sylvia Earle over 20 years ago. That led to Mission Blue Project. That led to becoming one of the 1,500 UN Ocean Ambassadors, and now a spokesperson for a coalition for the North Atlantic ecosystem, the Mesoamerican Coalition group that's about 25 NGOs, but there's 250 waiting in line. So just kind of networking, getting all stakeholders to come to the table from all sectors, all countries, because it's all connected, blah, blah, blah. So just the Pied Piper from the UN UNESCO Decade of Ocean Science guy. So I'm proud to be here and look forward to learning and then being able to connect whoever in our pool needs to get connected to whoever's in this pool. And I know there's going to be lots of connections. Honor to you all for what you've been doing.
Great, thank you. Laura?
Hello, I'm in CSIRO like Jody, and I'm working on antimicrobial resistance in aquatic environments.
Thank you. Nastasia?
Hi, I'm Nastasia Patton. I am a project scientist with Scripps and NOAA, working with the Cal Coffee Fisheries Survey. We're trying to incorporate eDNA into our sampling protocol. I also am affiliated with the Southern California Coastal Water Research Project, or SCORP, that has their own eDNA sampling program. And a big part of this affiliation is to align our methods and approaches to eDNA sampling and processing and analysis. And so I've kind of been loosely affiliated with BeBOP for a while. I haven't contributed very much, but I'm very excited to start using the resources that they've built, and hopefully can help out by doing some of that like beta testing and feedback. So yeah, really excited to be here and interact with everyone.
Thank you. Laura? Oh, sorry. I'm going backwards, aren't I? Nick?
Yeah, hi everyone. This is Nick Jeffrey from Department of Fisheries and Oceans, Canada, for Federal Science Agency. My team, I'm based in Halifax, Nova Scotia. My team works on marine protected area science, and we use a lot of eDNA metabarcoding for developing time series of monitoring marine biodiversity, as well as a lot of population genetics type projects, genome sequencing and SNPs and so on. And yeah, just interested in moving towards some form of standardization, or at least harmonization of methods, especially for eDNA. As a department, we have many research groups that use eDNA in particular, and we all do things a little differently. So working towards that with this group. Thanks.
Right. Thank you. Yasmina?
Hi everyone. Sorry, it's quite a wee morning here for me. My name is Yosmina Tapilatu, and I am a marine microbiologist from Center for Deep Sea Research in the National Research and Innovation Agency in Indonesia. And I recently just started in, I'm a marine microbiologist, so I just start to study the bacterial composition from the deep sea area in eastern Indonesia. And I am interested to see what kind of standardization in terms of molecular biology that I can apply in my work. Thank you.
Great, thank you. Okay, I think I'll just go ahead and start the presentation. Many of you are very involved in this, so feel free to jump in or follow up. I'm going to start, and then Katie's going to pick it up. And this is a small group, so anyone has any questions or any comments or suggestions, just go ahead and raise your hand and go for it. I need to share my screen. So this is basically a presentation that Katie put together, so I'm really grateful that she did that. She gave this presentation essentially to the OBON group a couple of weeks ago. We had it as a poster at GeoBon, so we're sort of moving around trying to get it out there. And I was kind of trying to remember the history of where we came from, and this is what I think, and you all can tell me if I'm wrong, that in 2021, there was a, let's see if I can see all your faces so I can tell if you're nodding, if you're going in and out. There was an OBPS workshop on eDNA and marine systems, and several of us who were there, as part of the follow-up, one of the questions was, what would really help? What would help to move eDNA and marine systems forward? And one of the responses to that we felt that we could potentially tackle was, if not standardization, at least a repository, a place where we could, A, put our protocols where they would be citable and findable, and B, find protocols. So from either the people who are already doing work and people who are coming in looking for protocols, an easier way of sorting through things and finding things than the current method, which largely seems to consist of calling people you know. So trying to get a little bit beyond that. And then a group of us, various groups have kind of come in and come out over the years, but quite a few people met. We talked about that and really tried to think about, how do we compare Omics protocols, practices, in a way that allows us to search protocols. So instead of just finding everything there is to do about the ocean, saying, how do we sample in a specific environment? How do we sample for a specific species in a specific environment? So are there ways that we can get into the protocols and make them much more accessible for people? And there's just, as we all know, there's so much Omics work out there, right? So those of us who are at GeoBon or just at the Marine Workshop, in California. There's just tons of work out there. And the more work there is, which is great, it's great that there's more work, but the harder it is to find protocols. So the more important it is to be able to organize them. So I think this was 2022, if I'm getting the dates right here. We applied for and became an official task team under the Ocean Best Practices. And under that group, or as part of that group, we developed a paper. This was Robin Samuels, the main author on that. And I'm going to, whoops, no, I can't do that. Next time I'm not doing a presentation, I'll copy this link and stick it into the chat if anyone wants to get to this. So this was just a general paper describing what we wanted to do. And we put quite a lot of work into thinking about what are the different components of an omics workflow and set of protocols that we would like to be able to break out so that people could look at things based on components of a workflow, not have to look at the entire workflow. And so, oops, actually there was one more step to that. So I'll come to the workflow in a minute. Then shortly after that, maybe this was 2022, we also put in a proposal to be an Ocean Decade project under OBON, which is the Ocean Biomolecular Observing Network. I can't tell, these people are in my way. So this is a list of OBON hosted projects, and this is Better Biomolecular Ocean Practices, which is BeBOP, is one of these Ocean Decade projects. So George, I think you said that you were involved in the Ocean Decade, so this is officially a project under that umbrella. And Katie is the lead on that, so it's led through Barry. Katie's the lead on that, and that's a proposal to sort of really try and work on getting a home for protocols, a structure for these protocols, and a way to encourage people to start uploading things. So when we thought about the protocols, we spent quite a lot of time thinking about what are the different components that people should be considering, that you might have a separate protocol for, or that you might have as components within your protocol. So we looked at everything from early on, what's the question that you're asking, what's your design, getting your permits, all this sort of pre-planning stuff, to the actual DNA workflow, sample storage, QA, QC, where do all those components fit in that workflow? And then we sort of modularize that in terms of different components of where we are. So there's a society part, which is really around what is your question? Is your data coming with FAIR principles? Designing logistics, which is the early stuff. The things that many of us think about maybe a little bit more when we think about omics, which is this sort of workflow, the sampling collection procedure, and then the kinds of metadata that we should be having associated with each of these steps in the workflow. So thinking about the metadata, we were thinking about the minimum information about an omics protocol, so MIOP terms, and for each of the different methodological categories, society, design, logistics, etc., we came up with areas or components that there should be metadata about. What's the project? What's the purpose? What analyses are you doing in terms of the environmental context? Where is it? Broadly, more locally, what kind of medium are you working in? What are the requirements? What kind of skills do you have to have? What kind of equipment do you have to have? And as a sort of a follower, how do you track where the protocol is, who created it, what language is it in, what was it issued by, how mature do you consider it? So ways of information about the protocol itself. So these were all the minimum information that needs to go with the protocols that could be potentially uploaded into the system. So then we spent some time thinking about what is the best way, how can we make a system accessible that people can upload data into? So Katie created a GitHub where you can upload information. There are a bunch of advantages to the GitHub. You can track changes, you can get a DOI, so you can get credit for your work. It's free and it's open access. And you can link to the open data and information system. And this is just an example of what one of those pages might look like. And I think I'll just go through to the protocols.io, and then Katie is going to walk you through what these pages might look like. So they're markdown templates for different types of protocol collections, and there are a couple of collections in there now. So Katie's put in the MBARI protocols. I think Jody, did you put some in? Are some of these yours?
Race
Sure.
added some from Fram, and I think another Pierre may have added some too from his project, but we're still in the beginning stages of adding protocols in.
Yeah. So we have some protocols in, and we're sort of in the part of the conversation we're going to have later in this is how to help people to be able to upload into this and also to be able to access it. The other place that we're considering putting these is protocols.io. So one of the questions we'd also have for potential users is it easier for you to access if it's on one platform versus the other? So these are more examples of the markdown templates, and Katie will show you that in more detail, so I'm not going to go into it. And then protocols.io is another potential home for these. So that's the sort of the background and the structure, just trying to find a place to put things that are easily searchable, that can be found, that can be credited to the author, and that we can use to create, I wouldn't say standardization is where we're going, but at least best practices that people are using protocols that have been used by, and to some extent vetted by other people who are doing work in a similar area. So I guess, are there any questions on that part? And then Katie will sort of walk through this process. Any questions or thoughts on sort of how we got here?
I'm impressed.
Good. Thank you.
Yep. And I forgot to mention, I'm also one of the original ocean best practices geeks. So I'm kind of wearing that hat today too, best practices. So this is much like what's been being talked about. You all are way up there ahead on the curve of where we need to go and get. So kudos to you.
Great. Thank you. All we have to do is actually get protocols up there. Then we'll really be on the curve. But
well, I can jump in and start sharing my screen. I just wanted to add too that something that was really important to us as well is being able to find out what protocols are in use currently, like today. Because I think one hard thing about looking at published papers is that there's often such a delay between when the samples were taken and processed and when the paper was published. And so I think it's really important to try to have a place where you can look at, what protocols are in use right now by these different programs. Because I know at least within our sampling program at MBARI, we're often having this, our methods for environmental DNA, they're changing so swiftly that it often isn't reflected by our most recently published papers. Sometimes it's changes that are happening within the last year or two that haven't been published yet. So this is kind of a place where you can share what you're doing right now too. And part of that ties into the maturity level of the protocol you're sharing. But here, so I'm sharing my screen now, and here I have it just on the GitHub homepage for BeBOP. And so I don't know how many people are familiar with GitHub, but it's a place that was developed to share code. But what's nice is it's free and open access, and we can create these kind of folders within a GitHub page that are called repositories. And so as part of the BeBOP project, we kind of had this two-targeted approach where we wanted to develop metadata that can be used to describe a protocol that would help let you sort and search through protocols. And then we also wanted to create these modular machine-readable templates that are those kind of boxes of how we split up the Omics protocol chain, so that it's easier to compare someone's extraction protocol with another person's extraction protocol, and it wasn't all together in one large document. So just to show real quick, for example, our MIOP folder. So we have a little bit of description about what MIOP is, but here is where we worked to define the different terms. in a certain computer language that actually is the same as what the Genomic Standards Consortium is using for their MIXS terms. So we kind of see these metadata terms, which we're defining here, as being eventually hosted and aligned with how GSC hosts their MIXS standard, which is minimum information about any sequence. And so we're hoping to have the work that we've done to define these metadata defining protocols also be part of that GSC collection. And I don't know how many people have used the GSC's MIXS standard, but it's something to keep in mind when you're depositing sequence data into the INSDC or NCBI GenBank resources. And so this tool has also been nice for working together as a group, where within GitHub we can make changes together to documents and then also post issues. It's a nice way of collaborating. And so here's our folder that has our protocol collection templates. And so here we've spent time creating these machine readable documents that when they're formatted, you can export as a PDF if you want. But what we wanted to do was create a location where you can share the MIOP terms, those metadata terms about your protocol, but then also just organize all the information so that it's easily comparable between groups. And so that's why we created kind of these different sections and tables. And so, for example, this is the PCR template, and under the standard operating procedure, we just thought, here's where you could share which PCR primers you're using, what are the PCR steps that you're using. And so just trying to format that information so that we can not only compare PCR protocols between different labs, but then as your own lab is changing protocols, you could be able to track changes within that document. So when it comes time for people to share their own protocols, they can what's called fork or copy this folder of templates and then edit the templates to put in their own protocol information. So I think I can just share. Let's do the 18S CR.
So do me a
For
favor,
example,
do your 12S touchdown, because I have questions about it.
let me see how much this one. So I'm also still in the process of putting the information in here, but we just have areas where you can link if you've already, so this protocol we created a long time ago, a protocols.io protocol about it. So we're trying to create links here of if information exists in a paper or something, you reference that, talking about, the method description, some of the equipment needed. And then here I started filling in, what our PCR primers are, the sequence of those primers, the protocol. So yeah, the nice thing about this kind of markdown language is that, like you're seeing on this screen, you can export a version that's or a PDF that's very human readable, but it's also machine readable too. So if a machine can go through here and be able to read the text, which isn't always the case with like a PDF, for example, it's harder to compare document to document.
So Katie, can I stop you there with a question?
Yeah, sounds good.
So, for instance, we've been working with this protocol and we've messed with it because that's what we all do, right? So if we wanted to put our protocol up in a way that people could really easily say, this is the Embari version, this is UNH version, what's the difference between them? Is that, how would people, because they're going to wind up with two MyFish touchdown protocols and they're going to be like, so what's the difference between them? How do we differentiate?
So the easiest way, I think, is to basically, they call it on GitHub, they call it forking, but it's like you're creating a copy of the document and then editing it. And then within GitHub, you can look at the exact changes that happened to your version. And then you can create a DOI for your version. So then we can see that your version came from our version in the beginning, but you've made these changes. And you can, when you're making the changes, you can put a comment saying, we changed this because of our environment and we needed to do blank. So I think, in a minute I'll show on protocols.io too, but both GitHub and protocols.io have the ability to track changes through time. If we start from the same document, then you can look at versions of that document or look at where people have made a copy of that document and then made changes downstream. So I think that's the advantage of using these both GitHub and protocols.io.
Yeah, and I just think that's super important because most of us don't create protocols from nothing. We take somebody else's and we mess with it. And
Right.
if everybody publishes separately, you wind up with this just morass of protocols that are really only subtly different. And if you can link them so that you can understand that they are really subtly different, and maybe it doesn't really matter which one you use, I think that that's going to be, it's going to help narrow down the field of potential protocols a lot.
So let me switch over real quick to protocols.io. Let me just show, so here on protocols.io, this is a different online tool that you can sign up for a free account with. And we've created a little workspace that we're calling Better Biomolecular Ocean Practices BeBOP right now. But we've kind of just started using this tool. And I've put some examples of protocols on here. What we've done is taken our templates that we developed on GitHub and then also imported them into this space so that, for example, if you look at the PCR one again, it just has those different sections that I just showed on GitHub. But here, protocols.io might be a bit more friendly for people if some people are already using protocols.io and can just already edit these documents. But one advantage to protocols.io is that it already has a feature we can chain protocols together. And so I wanted to show an example of that. So it allows you to create these protocol collections. And so one goal of ours was to be able to create these modular machine-readable protocols. So you can go, just like in that figure we showed you, from sampling to extraction to PCR to sequencing, like your whole workflow, and then be able to compare within those chunks, but then also say, for this data set, here is the whole workflow that we did that resulted in this paper and data set. And so the nice thing about protocols.io is you can change those individual protocols together and then create a unique DOI for that chain. And what we really like about this is that some people might, be using almost the same protocol chain as another group, but then just changing, for example, the extraction protocol. And this allows you to swap out, a protocol in those chains in one, like the extraction protocol in that chain, and then get a new DOI for it. But it allows easy comparability between workflows between different groups. And, to get back to that, how do you track changes in a document? I wanted to show a quick example. If you open up a protocol in protocols.io, you can also look at changes, and it'll go through and highlight. Here's a Mbari one that I've been working on. It highlights where I've added information. So it shows you highlighted in yellow where things have changed, like I've changed how those boxes were formatted, and then doesn't highlight areas where there haven't been changes. So just like in GitHub, you can look at how a document's evolved over time. And if you feel like this new version should get its own DOI, then you can also issue its own DOI for that version. So you can cite that version with your data set. So together we've started to use these two tools to implement both having metadata about protocols that'll eventually let us be able to sort them and search for protocols, and then also having these modular machine readable templates that lets you better compare protocols within this omics workflow. And so that, when we first got together, we identified challenges of, for people just starting to do eDNA sampling, like how do you find and sort protocols? And so with our MIOP, we can say, it lets you know we want a protocol in a marine environment, maybe applied in a certain area of the world, and you can be able to sort protocols by what you want. And then with the modular comparability, we can start to look at, well, what is the landscape of who's using what PCR primers within this group? And so right now, as a project within OBON, the Ocean Biomolecular Observing Network, we're working on gathering all the protocols in use by members of that network. So we can start to say, okay, here are the major initiatives and projects that are working with OBON. Here's the diversity of omics protocols that are in use around the world by these projects. And right now we're in the process of trying to gather those protocols. So here I can stop sharing, but does anyone have any questions on what I just showed? I know it's a bit of a whirlwind.
So just one thing that if like me, you find all those acronyms and GitHubs and very daunting, we are working on, I think Katie said this, but just to emphasize it, the ability to machine read a protocol. So if you have a written protocol, it can write it into this format, so you don't have to go through it in as much detail. So we're hoping that will really help people, but I guess Katie tried it. It's not quite there, but we hope to get it there. And then in a fantasy world, it's all machine readable, and you can go out and crawl the internet and find protocols and feed them in.
So as part of the UN Ocean Decade, we're hoping to be able, through that, I think in one of the slides we mentioned that protocols can be visible through this UN Ocean Decade data OTIS system. And so we would hope to have one spot where you could say, I want a protocol about DNA extraction for eDNA, and then be able to see both in BeBOP and then potentially getting OBPS hooked up to that system too. You'll be able to see where the different protocols are that are part of your search query. Yes, Jose?
Yeah, I mean, a very interesting presentation. I'd like to ask a very practical question. I mean, I'm the coordinator of the Blue Mission Atlantic and Arctic. I don't know if you know about the Mission Ocean and Waters that has been implemented by the European Commission. I mean, they are investing several hundred million euros on trying to create solutions that could be replicated and scaled up in the future. I mean, to restore our ocean and waters. I mean, in that the implementation of the mission is divided in 4 different basins. Danube is take care with restoration of freshwater systems. Mediterranean will be plastic pollution. Baltic and North Sea will be carbon neutral, circular economy, blue economy, and the Atlantic and Arctic is restoration of marine and coastal ecosystems and biodiversity. One of the questions that we have is always, I mean, to create solutions that will be later replicated and scaled up in the future, we need to assess if these restoration efforts are successful or not, you know. So, I mean, I was listening, how can we use this kind of, I know it's a step further, these protocols and all this, to assess the quality of these restoration efforts, so we can use this as a, let's say, as an example or a solution that will be later replicated in the future in other areas. Sorry for this very long question.
So I will say I just came from the GeoBond conference, and so does Nastasia, where there was a lot of talk about very much like that. So how can eDNA be leveraged to A, understand baselines, and B, understand change? So I'm not sure I can answer your question because there are so many different approaches. It depends on the environment that you're looking at. But there are a lot of people working on that. And one of the issues that comes up a lot is standardization, which is a slightly complicated term. So if you're going to compare what I did with what Katie's doing, if we're using wildly different methods, it's hard to do that comparison. Or if you do a baseline assessment and you come back 10 years later to look at that restoration and you use a totally different method, you're not going to be able to do that comparison. So something like this repository at least gives you access to the methods that were used. So if somebody says, I use method XYZ, you can go in, you can see that, you can see how it's related to other methods. There are other conversations going on about standardization, which this project is not really about that, but it's more about accessing and tracking the protocols that people use. So you could know what people did and be able to decide whether you wanted to continue with those particular methods, or if you're going to tweak them, what those implications might be. Does that help?
Yes, a lot. Yes, thank you.
I think a lot of time we talk about standardization or the need for standardization, and we feel this tool is kind of the first step of just increasing visibility of what everyone is doing. And so you're able to see across the landscape of what protocols are being applied where. As kind of the first step towards, okay, as a community, we now see what everyone is doing and we can have a better conversation about how we'd maybe want to coalesce moving forward.
So I might throw Laura on the spot here because I work with Laura on this antimicrobial resistance project. And I know, you know, one of the tasks she has at the moment is trying to figure out what everybody else is doing in the space in terms of methodology. How are they collecting their samples? How are they extracting them? What targets are they going for? And Laura, I was just wondering, after having seen the presentation, do you think if something like this was further down the track and people were actually putting their protocols into this format and on protocol.io or GitHub, that this would have helped with that task?
Oh, absolutely. It would be much more easier to just have a place where you can find all the information compared to screening hundreds of papers and then maybe not finding the information you need. So that would be absolutely a useful resource.
Yeah, okay, that's great to hear. And I mean, I agree. I've, you know, spoken to Katie about this recently. The marine microbiome work that we do, you know, we're wanting to extend that to larger organisms. So getting some metazoan assays up and running. And we've been trying to collate all that information about what all the other big observatories are doing around the world. And it's actually quite hard to find that information, you know, what amplicon are they using? How are they extracting? So yeah, I think it's a really valuable system.
So we do have some specific sort of leading questions. So if there are no general comments, we could kind of go to those and give you prompts and try and get you to help us out with some of the things we're trying to figure out about this. So I'm going to share my screen again. Is there other general questions or thoughts? Okay. I'm actually going to do this in this mode because I might have some questions, and that way I can type in answers. So this is where we were at the protocols.io, and just there are a couple more slides here that just show in a little bit more detail what all the different components that we sort of singled out were. And we don't have to go over them, but just thinking about all the different pieces that happen in a workflow, from all the way from figuring out what your question and your purpose is, to all the way to the end, publication and dissemination of your results. And every piece of those protocols should have some kind of, if not very standardized protocol, and not, you know, they should have some method that's associated with them. And all of those protocols could be captured somewhere. And then this was just breaking out that advantage of being able to search. So if you're interested in field sampling, you could search for field sampling, but you could also look for field sampling within a particular methodological category. So what are the resources that you have available, or what is the geographic location? So I'm interested in field sampling in Africa, and I have these resources. What can I do with that? So you can really kind of narrow in. Katie already said that, but that's just an example of where that goes. And I think these slides are pretty much just more of that same thing. And then this is sort of where we are with this project. So we've developed these templates. We're starting to map the workflows and the protocols of projects. So Katie was talking about bringing in the OBON projects. And then we're really working a lot in these two spaces here with the red box, getting feedback. So what is keeping people from doing this, other than not even knowing that it exists? How can we make it easier? How can we make it something that people are going to start actively using? So we came up with just some questions. We thought we'd try that and see if we could get just a little bit of feedback from here. So what groups are represented? So I know we have the Southern, what would you call yourselves in Australia? Would you be the Southern eDNA Society? Would you be?
Yeah, it is the Southern eDNA Society.
And I know you told me, so you know what, we can either, yeah, let's just go around and say this real quick. So Jose, you said that, and I'm not sure I caught it. Or you can type this, people could just type this into the chat and I could pull it out of there too. Jose, are they there?
Yes.
Yes.
Yeah.
So what group would you consider yourself representing?
Oh, I mean, I'm thinking how to reply to that. I would say that I'm representing the mission Ocean and Waters from the European Commission.
I know that's hard because we all have multiple hats.
Yeah.
George, you also told me, and there were a whole bunch of hats in there. So do you want to, sort of for the purpose of this conversation, do you want to pick a hat?
I just dumped it in the chat.
Okay, perfect.
Yeah, and you can put ocean best practices too. I mean, I don't know the exact purpose here, but that would be the three main things that I consider myself hats I'm wearing. The coalition, which is the whole North Atlantic ecosystem. You know, I mean, one project is 1,000 youth ocean ambassadors for 10,000 coastal communities by 2025. So,
Okay. I
it's,
know we've got Canada DFO here. Yeah, there's Nick. And who else? Vanessa? Or whoever?
And partly reason why we wanted to kind of ask this was because you can see in a later question is we wanted to figure out, you know, are there similar efforts out there that we might not know about, but
oh, yeah.
based on who's here.
How about the Yosema and Vanessa, maybe just pop it into the chat and I'll pull it out. Because yeah, for those of you who are practicing in the genomic space, what we really want to know is where are your protocols now? And I'm going to admit that mine are. Okay, so they're on the Google Drive, so the rest of my lab can read them too. But frankly, nobody else could get to my protocols because I just have not really been good about sharing them. I'm gonna leave this and come back to it. So, and I know that Katie's are on a website because I can look them up on the MBerry website, and some of hers are now in protocols.io. So,
But it's still a mix where I still have some new protocols that I need to get out there too. So it's always, yeah, it's always hard work to get them out there.
And not public, we'll just call that. Does anybody have a public repository where they are putting their protocols right now? So let's assume that most of us have them on their computer, but is there another repository? What we're sort of getting at is what do we not know? So is there another repository that we don't know about? Or is there another way of posting these things that we're not aware of?
You know, here, again, going back to the mission Ocean and Waters, we're developing a kind of catalog of solutions that will include also protocols. I mean, this catalog will be available by the end of November, beginning of December.
So that will include things like Omics protocols?
Yes, yes. I mean, anything, I mean, we call in the mission ocean and waters, especially on the Atlantic and Arctic Lighthouse, we call like solutions a unit of replication. So if this protocol can be replicated, they will be, I mean, included in our database of solutions.
And how would we access that? Is there?
It'll be open, and there will be I mean, they will be tagged, and there will be a lot of ways of searching for this kind of information. And basically, I start to discuss Jay and Renee. Maybe we can even create a kind of like a repository inside the OBPS, where will be these kind of protocols will be. We could find them easier for the international community. So we're still figuring out how to do that. But the idea is to have this kind of a catalog of solutions that will include from technologies for processes, from methodologies, and also protocols.
So that's super interesting. I think that's the kind of thing we need to know about, because that's going to be a repository of protocols that, yeah, if there's a way of linking that, then that would save double posting. I mean, are you familiar with this, Katie?
No, I hadn't heard of that yet. So yeah, that's great information to have.
Yeah, the mission Ocean and Water, I mean, they are developing what they call the mission implementation platform, with all the information will be like put in one single place, and then we have like subsets for each one of these basins, and we are taking care of the Atlantic and Arctic. And then I can share with you later a presentation on about this platform and how can we, let's say, make it interoperable with other databases as well.
That would be great. Yeah.
And by the way, this platform will be called Wavelinks.
So we do have people in this commission from Europe. So is that something they would know about, or would they have to be actively involved in that specific project?
Oh, this is a very good question. It was supposed to be open and visible to everybody, but you know, the mission is starting only now to disseminate all the products they are developing. So it take a while, but we expect next year to have a dissemination campaign when everybody will be, let's say, everybody will know about this platform and also how they can contribute to this platform.
Yeah. Yeah, that would be great. We'll check with Raisa and a couple other people we have from Europe and just see what they know about it. But
can I just kind of spitball something here?
Yeah.
So I mean, I have some microbiology knowledge and some DNA knowledge, blah, 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 biochemistry stuff, but there's several groups I'm representing now. And one of those is maybe well aware of, because they've been at it a while, out of Northeastern University, Ocean Genome Legacy. They've got like 25 to 35,000 marine DNA specimens in freezers and continuing to do that. And we're part of the group that found the recent coral disease thing. Then there's in Gloucester is a Genomics Marine Institute. And so, I mean, these are people that are doing obviously DNA stuff. And I'm assuming, there's actually even a Essex North Shore Technical School that their kids are doing DNA stuff in these. It's high school on steroids, but they were doing DNA while I was there just last week. So my point is, I'm going to plug them in. And I presume that, especially Ocean Genome Legacy out of Northeastern University, they surely have protocols and, and I know their intent is to walk the talk of of the UN and open, transparent. You know, that's how we're gonna, by doing all of this open, transparent stuff, we're gonna all end up with better science. So
Yeah.
anyway, I'm just throwing that out. Like I said, I'm just throwing that out there. I will share this group. That's what's my role is here, is to be eyes and ears. So I know enough to probably be dangerous, but enough to also make them get involved with you guys, and hopefully it'll lead to some sort of win-win for the planet.
That would be great. So in spite of the fact that I can't spell it, I do know some of the folks at Gloucester Marine. I just cannot at the moment figure out, how do you spell Gloucester?
GLOUC Gloucester, GLOUCHESCH, yeah, ESTER, yeah, there, Gloucester.
That's not right. Well,
That's
anyway,
okay.
we know
Yeah.
what we.
Yeah, and then Ocean Genome Legacies at Northeastern University near Boston
Yeah,
area.
yeah, I'm in New Hampshire. They're close to us too.
Yeah, and so there, and you know, well, there's another Ocean Alliance, and it's also Whale Org is the Snotbot guy that's doing doing eDNA stuff too. I believe they were doing DNA by flying a drone over whales, catching the snot, and then analyzing it for whale health and all of that, but also the DNA of it. But now they've got these two new darts, one that's a video camera dart that they can place exactly on the whale where they want, and it goes around for a couple of days, blah, blah, collecting video. And then the other one that goes down and literally takes off some skin and stuff like that, and monitors the depth and pressure and all that other stuff data. And then those two darts release and they pick them up and blah, blah. But that's all DNA stuff too. So I'm going to, the point is, everybody that I come in contact with now, because I'm promoting ocean best practices, of course, I will point to this group, and hopefully it gets it spread quickly, because that's what we need. Helps.
That is awesome. So Katie, what do we have by way of a link that we can send out? So we have a link to the GitHub.
Yeah, I'm wondering if we can share, because I did a little tutorial video too, so I'll see if I can get that up there.
Oh, you know, if you want to make it real easy, you guys just make one common email and send it to me. You've got my email by the registration, I think, or I'll put my email here in the chat. But just, yeah, just send an email and say, here, just connect me to Ocean Genome Legacy and North Essex and yeah, all of them. Anybody that I can think of that's remotely connected to genetics, DNA, because everybody's obviously very excited about eDNA now too. Extremely excited.
Yeah,
Here's my email in the chat too.
Yeah, we were trying to be a little bit careful not so it didn't, I don't know, we had this conversation about, is it just eDNA, but not just metabarcoding. So trying to be inclusive of other
Right.
more complex types of analyses.
Yeah.
Okay, that is super helpful. What I'm trying to figure out, I can't talk and move things around at the same time. So when we're done, I'm going to take a little time and just move this stuff in the chat into somewhere so I can get to it. Or will I have access to that chat after this ends, Jose?
I think we should be able to save the chat.
Yes, of course. And you can save it as well.
Okay, yeah, I'll save it because I think there's a lot of useful information in there and you don't all want to watch me cutting and pasting. Awesome. So this kind of segues into our next question. Would this protocols BeBOP be useful to you? So some of you may not be, so like George may not be actively generating protocols, but it sounds like you're connected to tons of people who are. For those of you who are generating protocols, would you use something like this? And I guess, what are the barriers to using it? So I gotta say the barrier for me is just it's a little bit time consuming. We all have plenty of other things to do. It's a little bit daunting, even though I supposedly know how it works. At this point, it's definitely, there's a learning curve to figure out how to get your stuff up there. Then there's the time to commit to doing it. And I think for me, just personally, there's also a little bit of fear of putting yourself out there when you always know that your protocols will be better if you just tweak them a little bit. So that's my honest personal assessment. And I'm curious how other people feel about putting their protocols up and what barriers you face.
I guess just to jump in real quick, even before BeBOP got started, we had, started putting out some of our protocols associated with DOIs, and it did really help with paper writing, that we could just say, and briefly in the paper, here's what we did and the main points, but then say, and here's a link to our protocol document that goes in detail of exactly what happened. And so I think when you get those protocols out there, then it becomes much easier to then share those protocols with other groups that ask you, and then refer to them for your published results. So even though it is effort, it pays off later on.
I would agree with you, Alison, on pretty much everything you said. We have, there's a smaller collection of protocols in terms of the IMOS work, because that's all very standardized. But when you look at the Australian microbiome in general, there are a lot of protocols, and fighting that time poor part of things in terms of sitting down to do it, because a lot of those protocols actually come from a lot of other institutions. It's trying to get them on board with who, they also have their own time poor issues to rewrite their protocols. And there's a lot of it because those templates are very information rich, which our protocols should be, but we all know they're not. It does also take a lot of extra time to add in the additional details, like getting the catalogue numbers for all the consumables and equipment you use. Really useful stuff, and we really should be putting that out there, but it does take time, and we really just need to, I think, just sit down and say, right, we're going to do this, and just try and knock it out, have a few people on board to do it, from our perspective anyway.
Yeah, I would just agree with everything that's been said about the barriers in terms of just time and sort of committing to one, even though it's not a commitment to one final version by any means, but I think it can feel that way when you put something online and link out to it, rather than, keeping it local and knowing you can tweak it at any time, and you don't have to like tell everyone, update it or tell anyone. The other, I guess, I think everyone's situation is unique, but for me, as I sort of talk to all of the different groups that are working on eDNA sampling in like the California Current or this environment that we're all trying to collaborate with, I think it took a while to actually reach a consensus on what might be the best 16S amplification protocol. And there's always kind of the caveats that we might decide on something and say, okay, this is what we're going to use, and this is what we're going to recommend other people use. But there can often be caveats where it's like, well, except if you're in this situation, then you can actually do things differently. And it's hard. I think it can be hard to incorporate that kind of nuance and caveat into a protocols.io template. Not that should be a reason to not use them. I think there are ways, I think, and you showed this, Katie, how you can actually add like comments and annotations, I think. So that's very cool. But I think it's just, I guess it's a long-winded way of saying there can be a hesitancy to commit to like one quote unquote final version of anything when it comes to sampling or molecular work, because those of us that do it a lot know that we're constantly trying to improve and make things better and tweak things. And so it's almost like, well, what's the point of putting this out there and formalizing it and doing all the work when we're just going to change it? But I think the type of version control that protocols.io offers, and the things like the annotations and the just the, you know, very streamlined like format of it, I think is actually it's going to overcome a lot of those barriers. I'm actually very excited to make a repository, put protocols in there, and once they're up, my hope is that it will be actually quite simple to update them, change versions, and be clear about what version reflects what work. So yeah, those are my thoughts.
Yeah, similar to what Nastasia just said as well. For me, it's, you know, there's always a little bit of fear about committing to a protocol, but like we've said, you know, you can version it, you can always tweak it a little bit, add a new version number over time, or create a whole new protocol once, you know, some people just will change their protocols over time as well. I personally really like the idea of BeBOP. The main thing for me has just been time as well, like everyone. But I think once you get past that sort of initial bit of time for developing your repositories and your protocol templates and so on, I think it will become quite a bit easier downstream. So, you know, I like the idea of having your protocols available, potentially for commenting on or citing, as well as versioning and having other people sort of contribute to them. I think another issue that some people have is that they do have this fear of something in their protocol just being blatantly wrong as well, and, you know, potentially being called out on or something, which in the end is probably a good thing to have something fixed. But I think there is a little bit of fear there too, that if someone's doing something differently than everyone else, it might be seen as you know, not accurate or not, you know, up to the date or something like that. But in the end, I think these are good things for sort of developing and streamlining our protocols.
Yeah, so Yosmina, I'm probably pronouncing your name wrong, I'm sorry. The comment about resources is actually super helpful, because I also think something that we need to be very aware of is that the people who are most likely to put up protocols are the people with resources. So those who don't have the resources and who have the protocols, the people without, you know, without maybe a big staff or without a big sequencing facility, the protocols that they need are going to be harder for them to find because they don't have the time to put them up. So that's something we should be aware of and try and figure out a way to help facilitate getting those protocols up there.
We also have to we should think about a lot when you're sampling under different conditions, like say you don't have access to a -80 or to different ways of preserving your samples too. So I guess working with different lab groups about what preservation techniques might be most useful when you're in different locations for field sampling. So having those kind of different options documented would help people who are planning sampling campaigns.
So would it help people, and just to be clear, we don't have any funding for this whatsoever, but if we were able to find a bit of funding and we had someone who could sort of help you, you'd say, okay, I want to do my protocols, you know, sit down with me on Zoom for whatever, for a couple of half days and help me do that, or do some of it for me. Would that help overcome some of these barriers?
I think it's always good to have something of a something to hold you accountable, like something that's put on your schedule and assigned a time, and you show up and are forced to think through the process. I think that's always helpful. And so and it's of course always great to have someone else show you some technical techniques rather than trying to bubble through it yourself if it's the first time. So I would say it can only be a helpful resource, something like that. Yes.
Again, I'm a caveman on all of this, except for I'm very familiar with corporate training and industrial training and stuff like that. And I was going to suggest what you basically have kind of just suggested on your own. I would just amplify it. Yeah. A few Zoom calls where you take someone's need to say, okay, I want to put this XYZ data in the system. And so you have a panel of 5 people there, and you kind of all interact as you fill out the form live and record the Zoom session. So now people have an onboarding, at least introduction to it. And if they watch that, then maybe they make an appointment with a now certified, and that's what you begin doing with people. You become a certified mentor of how to use this platform, and you're willing to take on one person a month or something like that and handhold them, and you begin to build up your little force of mentors, and you keep going until it becomes a ubiquitous thing, because that's, again, this is just going to be something that once people, I think it was Jeffrey, once people use this a few times, once they've put half a dozen projects in it or whatever, it'll be just like using PowerPoint or Word or Excel or anything else. It'll become, and they'll begin to have the benefits, which, if, again, I'm the caveman, but what I think I'm hearing is, if you do all this long term, the trade-off benefits that you're going to gain, that the whole eDNA world will gain by this being done, is going to be monumental. So there's all the wins ahead. Somebody else said it, Jeffrey, somebody, again, bite the bullet, roll your sleeves up, start doing it, and make you up a couple of examples, half a dozen examples of walking somebody through their entire process of taking their data, however they've got it, getting it into this system, and record that, and then people watch that, and if they need more hand-holding, they escalate them to the senior mentor path. And that's my 2 cents.
Yeah, appreciate that. Yeah, I think that is definitely helpful. Katie did a video.
Yeah, this last spring, I think it was, I just did a little tutorial, but things have changed a little since then. So I'm thinking like maybe, yeah, recording a little tutorial again that can be short and on the GitHub, and then also linked to on the protocols.io website, I think would be useful. What's nice about protocols.io is it's, I think, much more user-friendly. So I think that might help people a lot. And people are already using protocols.io too, a lot of times. So maybe a little bit less of a barrier there.
Is it an awareness of the platform then that's really the barrier?
Yeah, I think there's definitely getting the word out, but I think we need to get a first set of protocols on there. I think we'll work, with the OBON projects to get everybody's protocols in there so we can kind of have a demonstration of how useful it is. I think that's the next thing we got to target. And then I think when you can look up other people's protocols and see what everyone's doing within this network, then hopefully we can spread from there.
So maybe what I'm hearing is you're at a certain level, you're not ready for worldwide, let's go bring people to the table quite yet. You want to start giving that list perhaps, but you need maybe a handful of players to kind of help you take it, just ratchet it up, a little bit of input, a little testing, a little bit more data inserted into the thing. So you've got a little bit better mousetrap to show people what it looks like.
I think so. Yeah.
Okay. I get it.
And I think that's what we hope the OBON projects will bring, because that's a solid set of projects with a good spread. And I would also say there is a bit of a carrot. I mean, if you are one of the early people to have your protocols up there, then your protocols are more likely to be used because they're up there and they're available, and other people's are lost on their computers. So there is potentially that. If you're interested in having your work recognized and used, that's potentially a carrot.
Well, I can volunteer the West Coast OBON to be one of the first repositories. Yeah, I would love to have. Right now, we literally have a Google sheet with, each protocol is a row, and it links out it links out to either existing repositories or wherever else they are on the web. And so to turn that into a repo with nicely formatted protocols would be fantastic, and I think hopefully pretty straightforward. So yeah, happy to be one of on the vanguard of this, if you will.
Thanks, Nastasia.
I think that those were kind of the questions we had. Are there other thoughts and comments? We have a little time, but we don't have to use every minute of it.
If I may ask something, I mean, how would you pack this solution in a kind of, these protocols in a kind of a toolkit that we could, let's say, use it, adding, I don't know, manuals or capacity building or training sessions? How could you pack this in a way that will be easier to replicate and to get more people on board?
Well, I think one of our dreams of what this information could let us do is have something like a decision tree or some way to sort through all the protocols. So that's really also why we developed the metadata describing the protocols, so that you could say generate a map around the world and see where the protocols are being applied, or say I'm really interested in this type of substrate in this environment, and then what are the protocols that people have used to sample from that environment. And I think, also being, it's very important on the data set side of things too, to understand what exactly happened to produce that data. And so one useful thing of having a detailed protocol that you can just cite with the DOI is when it comes time to share our data in OBIS or GBIF, that we can just link to that protocol and somebody can discover the full protocol of how that data came to be.
We did have early on, when we were sort of envisioning trying to figure out what we wanted to do. Remember, Katie, there were discussions about things like videos. So you could upload your protocol and then also do a video, like this is how you do it. So it would be protocol and training rolled into one. It's just that is so time consuming. We backed off on that, but I could see a version down the road where there might at least be an option to put up a video. So if you have a protocol and you have a training video, or you really want people to be able to use your protocol, that might be an option. But
well, within protocols.io, and I think GitHub too, you can link to videos easily. So it's something where if you already have those things, then you can include it within your document.
Okay. If that's it, I'm going to save this chat because you all gave us some really great suggestions and I really appreciate it. You have both of our emails, I think, should be in the contact info, but I'll just drop mine into the chat here too.
Can you just paste that chat? Because I think that's an administrator can copy the chat. I don't think attendees can copy the chat, but you could send us all an email with that as the chat content.
I can. I assume, Jose, do I have access to the attendee email list?
Oh, I'll send it to you as soon as we finish.
Okay, if you send me that, I will save this chat. So I assume I can save this.
Yeah, somewhere there's a save chat administrator function.
Yeah, I just want to make sure that I have that power, but I will make sure I have that power before I sign off.
It's under, if you go to type message here with the chat, I see the 3 little dots to the right hand of that box, and I can click on those 3 little dots, and then I get an option to save the chat. So I don't know if everyone can do that,
Yeah.
if it's just hosts and panelists, but
Yeah, I can't.
okay.
Well, I not only can, I have.
All right.
And I'm going to get an email with the attendees, and we're going to send that out. And also, before we send it out, we might just take a little time to see if we can find, I will send you a link to the paper, or maybe just attach the paper, and Katie's little sort of introductory video about BeBOP. So you'll have a little bit
And
of
if
information.
this is recorded,
Mhm.
put that link in there too. And now we kind of have, in my case, I now have something to send to about 5 contacts and say, here, go watch this video and see if you don't think you guys need to contact these guys.
That'd be great.
Yep.
Be awesome.
Yep.
We want a big party.
Yep. Well, again, you just need a handful of other people that are in this space. What I heard, a handful of people in this space that can help populate, use this, do this thing, and you track it along and you find where your gaps are, try to fill those gaps. Meanwhile, you get something you can get out to the world and grow it to the next level. I get
Yeah.
it, I think. So I'll try to help.
Great.
Thank you.
Super
Sure.
helpful.
Anyway, good luck with it all. Honor to your work again. And see you at the other best practice sessions.
All right. So anyone else? We good? We're good. Okay. Thank you all so much for coming. We really appreciate your input.
Thank you, everyone.
Thank you.
Okay.
Bye.
Thank
Thank you.
you.
Bye.
Thank you.
Bye.
Bye.